I'm Rasmus Hansen. As Matt said, I'm the director of client development and project management here at 21st Bio. The part about project management, I think, stems from when we started the company, and each of us had like 10 different hats we were wearing at the same time. So project management seemed like a nice bucket of other things. These days, I'm using more and more of my time at attracting new clients and also running projects with clients. I'm originally a microbiologist and a European patent attorney. Spent 19 years at Novozymes before switching to 21st Bio. So I want to talk about manufacturing with precision fermentation. Let's see, slides are not changing. There we go. So if you look in the bottom left corner, you see the source here is McKinsey & Company Biorevolution in 2020. They made this report where they calculated that actually 60% of the physical input in today's world can be produced with biology. Of course, a lot of the stuff that we use is based on rock, concrete, steel, stuff like that. But also a lot of the energy and materials based on fossil fuels can actually be supplemented or even substituted with things that are made out of biology. We talked about some years ago in Novozymes that sugar was the new oil. You can say back then, a lot of the technologies were not mature enough to really make this transition. I think today we have a much better shot at it. You can say both there's more urgency, there's more supply chains that are under pressure, also in foods. And of course, the production technology on the biological side have evolved a lot since then. Not changing. So what we do at 21st Bio is we're providing technology for production of proteins with precision fermentation. So it's not our ambition to be a production company. We want to develop and optimize technologies for our clients, and then they need to go out and either establish production facilities or produce with contract manufacturers and take a product to market. A lot of those products will be B2B. Some of them will also be B2C. But we see ourselves as, you can say, not necessarily the very first part of the supply chain, because if you look on the left here, the molecules can either be something you take from nature. We have later on milk protein as an example, something you take from a cow or potentially a human as well. The molecules can also be invented or screened in large screening programs to find the molecule that has the best capabilities for some certain application. So then once you've found your molecule, we can help you actually make the production technology that enables you to produce the molecule economically feasible in large scale. And I think the core competences here is the strain development, where the strains are cloned with the genes encoding your molecule and optimized, and then the processes for fermentation and downstream development are being developed and optimized. And then lastly, I think we see a very important point in helping our clients transition to larger scale. As you'll see in a few slides, we've invested in pilot facilities here in Søborg, Copenhagen, where we are based, to enable customers to get to the first 1,200 liters, and from there we help them move on, typically to a CMO with larger volumes. For some of them also into their own facilities. But we've seen really that the scaling step going from the lab and into the larger fermenters, both there's been a lack of capacity, but there's certainly also a lack of competency to do that step. And it's something we see that it's quite often being underestimated how difficult it is. I think coming from the industrial world, we all know examples where even established companies, sometimes they even use 2 years to do a successful tech transfer of their own strain from one facility to maybe to a new one. So I think a lot of startups really underestimate how difficult it can be. Oftentimes it's related to, the process conditions you run in your lab or maybe even in your pilot. You cannot copy those conditions one-to-one when going into another facility. If you're going into a CMO, for example, that CMO is used for a lot of other things at the same time. So obviously, the conditions that the equipment need to be able to run over has quite broad ranges, but maybe it cannot accommodate, the specific ranges that you need for your process. Then you need to do a lot of optimization to find, okay, then what's the best alternative, and that's not necessarily easy, right? Sometimes you need to even invest in specific equipment at the CMO to really be able to get good enough performance. So I would say our core competence actually spans quite broadly here. In-house, we do the strain development, we do the process development and optimization in a scale up to 1,200 liters. And then we have the fermentation and DSP consultants that helps the client go into the CMO, do the tech transfer, and then succeed with the first runs in larger scale. Of course, that's where you start to commit a lot of money, right? When you run the larger fermenters, it's cost per run increases a lot. So you want to do that right as quickly as possible. So just briefly on the value chain here, we think it's quite important that to kind of include the farmers here. Of course, as I just said, we developed processes and strains and so forth, but really fermentation is based on agricultural output. Typically, it's purified sugar. That's what these processes have been developed for, because so far in history, even though companies like Novonesis have produced large quantities, it's still quite small volumes and quantities compared to what we are potentially looking at in the future. So going into lower value compounds like milk protein, silk protein, for example, we're talking about potential volumes that are much larger than what all the enzymes Novozymes has produced over the years. And there, of course, the carbon source is super important because these molecules are going to be competing at low cost. So you need cheap sugar sources, which means they need to be fairly close to where you operate. They need to be purified only to the level that's necessary, and so forth. So the farmers play a very important role. And I think one of the reasons we also want to highlight that is that when we move into milk proteins, for example, we want to emphasize that there's really no conflict with the farmers. We're talking about substituting molecules, not replacing them, because there's such a big demand for protein over the many coming years that we need still all the available production technologies running at full speed. So the biological input, both from us, from the farmers as well. Then there's a fermentation and purification side of it, which, of course, the CMOs play a large role here because they have established capacity. all the startups that are operating in this field, none of them have production capacity. All of them, more or less, need to start with a CMO, which is difficult because a CMO is never optimized to your specific process. So you'll never have the best production economy with a CMO, but it's what enables you to get into the market, start selling your product. But eventually, at least if you have a product of any significant volume, eventually you need to build your own facility, because that's where you can start optimizing and saving on your cost every year. It's definitely one of the most important parts of Novo Nordisk's success over the years, that they've been able to improve strains and processes year after year with about 5%. And you don't get those optimizations and savings if you continue operating with a CMO. You can say, in a nutshell, you don't have the same objective as the CMO, right? Because every time you do an optimization, you actually need less of their capacity. So they don't really have a big interest in helping with that. Then lastly, on the product side, a lot of the outputs are going to be powdered proteins, because that's a very nice and stable form. It's easy to transport. It needs to be probably resolubilized when formulated into products. It can be food products, for example, drinks and foods, where protein and protein functionality is tested or is needed. And then we have the picture here of the rolls of silk thread, which is a nice example, which is not food, but a material that is not really available from any other sources, because this particular one is spider silk, and spiders cannot be grown industrially. They eat each other. So the silk that you know from your clothing is made by silkworm. It's used to make a cocoon, not a spider web. So the properties are completely different. Of course, the products are produced by B2C or B2B companies, and then purchased by FCMG companies making food products, for example, or materials. So here we sit in the very early end of the supply chain, the value chain. We operate in quite a broad variety of fields, foods, in terms of volumes, and probably also potential right now. I think the most important ones, the milk proteins, are very important. A lot of startups operating in this field because it's so obvious. These proteins are good, they're nutritious, but they also come from animals that don't have a very nice sustainability footprint. That's especially true for the milk proteins, to a lesser extent for the egg proteins. Cell-based meats, it's not something we do, but there's a lot of ingredients necessary for the media, for cell-based meats. A lot of the ingredients are very, very expensive growth factors, for example, that are known from cell cultures in pharma. And there's a big need to produce those in cheaper ways that are maybe not pharma GMP, but still have the right functionalities. Materials, silks, for example, for textiles, can also be something like collagens, gelatins for personal care, compounds that today are extracted from animals, used both in biopharma, in creams, other personal care products. Then agriculture, where, of course, insecticides and fungicides typically are based on chemistry, which is not easily being degraded in the environment afterwards, and oftentimes also have much broader activities than are needed. now we are starting to see protein and peptides that are much more closely directed to specific pests, which can be either insects or fungi, potentially also bacteria, recombinantly. And then lastly, a fairly new field, which is very, very, very interesting in mining. We see that a lot of the minerals that are used for electrification, I think more than 90% of those are controlled by China. And for example, the Department of Defense in the US sees this as a major supply crisis. Actually, they don't like being very dependent on China for these types of minerals, including the rare earth, that are very, very important for the electrification and battery technology. So the DOE is actually, or the DOD, Department of Defense, is investing quite extensively in technologies to extract minerals from spent mines. And some of these technologies includes peptides and proteins, which for some reason can bind to these minerals. I think it's probably based on microbes that are trying to defend themselves against these toxic minerals, and then they produce peptides that can bind them, probably to make them less harmful. Yes, moving on. So this may be quite well known to you, but I want to mention it anyway. So there's a lot of challenges of scaling protein production, and whereas you can say the very first part of the chain here, the capabilities of making molecules, designing new molecules, that has been democratized, so to say, right? You can do a lot with in silico screening. You can do computer modeling, come up with new proteins and other compounds that has some specific applications that are improved compared to what was known before. Screening robots are being more accessible and can do high throughput screening of lots of protein variants. So that part is quite easy. But the challenge is that you can make a lot which works in the lab, right? But the difficult part comes when you want to upscale it to like the real life. And one thing is the microbial strain platforms. So a lot of the work done in small scale is based on university strains or public strains, which are not really optimized for producing at large scale. At the same time, you have access to molecular tools that are public. They can be pretty good, but again, not really made for upscaling. Lastly, the competences for actually taking technologies into large scale, typically they've been sitting in the big companies, and they've not been available broadly for smaller companies. I think that's beginning to change. I think we see more senior people from the big companies are spreading out into the startups. But I think for some years, after maybe 2020 and a couple of years from there, this part of the journey was really, really vastly underestimated, how difficult it is and how different these competences are compared to what you know if you've only worked in a lab. And I think that was kind of what actually was the basis of what our founders saw when they kind of decided there's a need and we can help meet that need. So what they wanted to do was basically accelerate the world's quest for these sustainable foods and materials. Over the years, been a lot of good innovation that really app and the upscaling. And one important part of that was you can make a nice strain to produce your protein, for example. But if you're not able to reach productivity levels or tighter levels that are going to be economically feasible, I mean, then you're going to fail. And they saw that the strain library that they have at Novonesis, the former Novozymes. That this library was capable of making production strains that could deliver productivity in kind of an order of magnitude better than what you saw a lot of other places. So the idea really was to bring this technology to be available for other companies outside the core areas of Novonesis, which of course are the enzymes. Today, I think we are approaching around 60 employees in Copenhagen, Denmark, and in Davis, California. And we are accelerating this with two different models, I would say. So one of them is strain development for specific proprietary compounds. So, for example, if you're a company and you've done your protein screening and you've selected a variant of some molecule which is now more thermostable, or it has changed functionality or something, you can file a patent on your compound. Then we can make a strain specifically for your compound. It's exclusive to you because you own the compound. It allows you to go into the market and sell your compound exclusively with good production economy. You need to be able to control the application testing, application development, formulation, everything that's downstream from producing the molecule. But we can enable you to produce it at commercially relevant production economy. In the other model, there are some proteins where we've said these proteins have so large potential that we want to make them available to a lot of companies in parallel. And the first one we selected was beta-lactoglobulin, the biggest protein in whey from cow's milk. So we developed a production strain for BLG, beta-lactoglobulin, and we developed it to a certain point, and then we offered it out in the market to customers that needs to do the scaling, do the application development, and also do the downstream process development. Many companies actually want to do their own DSP to give the molecule specific properties that are different from others have. They want a patent around that to kind of mark out their space. Other companies are focusing on application development to make products out of the protein. But I think the main reason for offering this program, as we call it, is that the potential volumes of the milk proteins are so large that it's going to take a lot of companies to cover the field in combination, and likely some of them are going to fail as well. So there's definitely enough space for everybody. We are working on a new program with the caseins. So caseins being the major proteins in milk that are particularly used for cheese. And we are starting to offer that into the market as well. And the last point here, fast to market. The important point being that the strains we have from Novonesis have been approved both in the US and in Europe for production of food enzymes, meaning that they are well known to the regulatory authorities, adapted over many years so that potential toxins and problematic side activities have been removed, which means that it's much easier to have them approved by the regulatory authorities. It's quite easy to get the GRAS notification in the US. It's certainly not the same in Europe, regardless whether your strain are known to them or not. I think that's a whole separate discussion, but that's one of the points in this industry where Europe is really lagging behind. So an example Substituting milk protein. Milk consists of about 3% protein. I think there are around 200 different proteins in milk. The major ones being the caseins, which constitute around 80% of these about 3%. The whey being, I think, the biggest protein in the whey is BLG. But obviously, you can see that of the whole content of the milk, the protein is just a fairly small part. But these proteins are very, very nice. They have nice amino acid constitution. They are very good nutritious proteins. And therefore, it makes a lot of sense to try to replicate them with another production technology. Yeah, with an outlook to 10 billion people to be fed, it's quite clear that with the current production technologies, we are not going to be able to supply enough protein. Especially protein produced by animals take up a lot of space. And of course, if you talk about cows, they emit a lot of greenhouse gases, especially methane, which is not good. If you look at the LCA of producing milk proteins by precision fermentation, it's quite clear that you can actually, on more or less all parameters, reduce the input very, very significantly. If we look at the water use, the reduction is more than 95%. Of course, because cows live a long life, drink a lot of water. A lot of water goes into also, processing the milk. So here you just do that much, much more efficiently. When you talk about the land use, I think it's 60 or 70% of all agriculture is used to produce feed to animals. So obviously, if you have much, much more efficient conversion of your biomass to protein, which a microbe can do, you can save a lot of land. And that land can either be used to you can say, capture CO2 in trees, for example, or it could be used to produce other crops that humans can eat. Greenhouse gas emission is very, very large, especially because cows burn a lot of methane. So a potential huge savings there. And then also a very, very nice savings on energy. Lastly, we wanted to brag a little bit about this, again, the self-affirmation GRAS. So the GRAS process, you submit to the FDA in the US your dossier. In the dossier, you describe your production process for the way you make your compound, and you argue that it's safe for a lot of different reasons. In our case, we argue that it's safe because the strain has been used for a lot of other approved food products. It's a strain which is actually found in food products in Japan, for example. It's a fungus called Aspergillus oryzae. So even the strain itself can be used for food. And then the protein we're producing is more or less identical to what cows are producing. And of course, then we know that humans have been exposed to these compounds for many, many years. So now we've got what's called the self-affirmed GRAS status in the US, which allows our customers to actually go on the US market and sell the protein. That's not happening yet because none of them have scaled to a size where they are actually entering the market. But we expect them to do that during 25. So that was my last slide. Matt, you said a short round of questions now and then a longer round of discussion later.
Sure. Yeah, I think for the sake of time, we might move on to Kelly
Yeah.
and then just do
Okay.
one larger at the end.
Yeah,
But thank you
that's
so
fine.
much for this informative presentation. It's really fantastic. I know I personally have a ton of questions and I'm really excited about the work that you're doing. So thank you so much.
All right, I'll stop sharing.
Okay, I'm going to share my screen here.
Great.
Okay, there we go. Are you seeing the slideshow?
Yes, we are.
Okay, great. So I just have a few slides. They're not nearly as professional as the ones that Rasmus just showed us. So, but I'm going to give a little bit of background on myself, a little bit on some things that I learned about scaling up, working with industrial products at AgBiome, and then some general needs that I perceive in the industry, and a little bit about what Sable Fermentation can offer, which I'm working with them as a consultant right now. So. So let's see how do we do this efficiently. There we go. Okay, so short little bio for me. I have degrees in chemical engineering and environmental engineering. I have started a company called Pastoria Bioscience, which was a biopesticide company. Sold that to Syngenta. I did work at AgBiome for about 6 years, which was a local biotech company here that produced biopesticides and also traits for crop plants out of a very large library of novel microbes. And I went from there to a company called Join Bio, which is up in Boston, and they were producing biofertilizers, which were engineered microbes for that. I was the VP of R&D at, there's no name on that one. All right, that's DMC Biotechnologies, which is a local company here. They have a lab here in Morrisville, North Carolina, near RTP, and another one in Boulder, Colorado. And then the last one there, which also the name didn't come through. Anyway, slide translation not always working. That is the logo for Sable Fermentation, which is a new local CDMO for fermentation and downstream processing here. So I have a lot of background in producing biological products, doing the process development, doing the scale up, the registration, and the actual commercialization of products for agriculture and for industrial production. And so some of the things that I've learned from what goes on with these small companies, and this applies not only to AgBiome, but to all of those other companies I work for, and Rasmus already alluded to a lot of this, finding tollers to produce even relatively small pilot batches up to 100 liters can be very challenging. There aren't a lot of facilities that will do that, especially for non-pharmacological products, non-human pharma. And so people need to build in more time for that, right? This is the thing that I think trips up a lot of small companies, is not leaving enough time to deal with some of these issues. And the next one then, of course, is getting the permits, right? The regulatory regime, of course, is different everywhere you go. In the US, it even varies state by state. And so you have to get permits at a national level and a state level, and sometimes even a local level, even to just move your microbes around. Much less to produce them at larger scale and then to release them into the environment if that's what the product is for. So people need to build in more time for that. You need to build in time to get the operators at a contract facility or a toller familiar with working with novel microbes. This is going to be the case with a lot of these novel industrial processes too. Even if your host strain is E. coli, the engineered strain that you're bringing to them from a process like something that 21st Bio would do for you is not going to be familiar to that toller. And so they need extra time to understand how to handle that microbe, what its specific quirks are in their process. And as Rasmus alluded, every facility is slightly different. So it's not going to be optimal for your microbe, even coming out of a professional optimization process like 21st Bio is going to offer. So you got to build in time for that. Your tech transfer pricing is going to be very important. As for a small company, you have to understand, a toller is going to charge for their time to start off with. And so you want that to be a separate body of work from the actual manufacturing, and moving to an outcome-based pricing. That's what's important for a small company. And as was previously discussed, you as the producer, your incentives are not the same as the toller. The toller wants to charge you for as many runs as possible. You as the producer want to produce in as few runs as possible so as to optimize your production costs. So that's a negotiation that people don't often plan ahead for, or think is going to take as long as it does, or become as contentious as it can. And it's just these are just a few things that, I've learned in doing a lot of this, that's just going to be very impactful, all of these things, as we move into a more bio-based manufacturing economy. And then some of the other things that Jen suggested that I should discuss with all of you today are general issues that have come up in the field of biomanufacturing. And so again, some of this we've already alluded to in Rasmus' talk, but there really aren't that many options at the pilot scale. So 500 liters up to about 25,000 liters, it's very difficult to find a facility that will produce at those levels. And the facilities that are available are, in high demand, right? So it can be challenging, and that's the scale, up to about 25,000 liters that a small biotech company, especially for agriculture, is trying to produce at the kind of right at that level to do their initial field trials, their registration trials, their sort of regulatory process. They don't need 100,000 liters of material, but it's often difficult to find this intermediate scale fermentation. And we already heard that every process needs different downstream processing. So a toller may or may not have the right equipment. It may not be similar to what you've worked with in the lab. It may not translate immediately, depending on how your microbe behaves. And so you might actually have to go to another facility for that. And I experienced that numerous times at different companies, where we could find a fermentation toller, but then the downstream processing, either they didn't have the right equipment or they didn't have it on the same site, having to then, contract with yet a third company to go actually do the downstream processing, especially drying. A lot of fermentation facilities don't have extensive capabilities for drying. And so that can also complicate your biomanufacturing process. So as we're thinking about, developing all of these bioeconomy type infrastructure, it's important to think about DSP in addition to fermentation, right? How are we going to enable that? And then I didn't even get into formulation, which is a whole other can of worms that you can get into after you've done all of that. And then the other thing that happens when you're scaling up is, of course, that your supply chain is not optimal, right? Your raw materials can be hard to find in those exact intermediate quantities. It's easy to buy, 1 kilo or 5 kilos from Sigma, but you don't want that unit pricing when you get up to trying to produce even at 25,000 liters. And yet some of the raw material producers are not used to selling anything at less than tank car volumes. And so, getting your supply chain to match up with what you need to produce can be challenging. And high unit costs, of course, drive up the cost of the final product, and that's not what you're after. So all of those are issues that have come up for me. And then this slide is really just about the digging in a little bit more into the lack of some of those resources. A lot of the products that we're talking about, these industrial products, the things that the McKinsey study showed can be produced by biology, often can be difficult to find a place to do it, because most of the facilities, if they're geared for pharmaceutical production or certain other kinds of products, if your product is non-pharma and non-food, they don't want to handle it. The two regulatory regimes, especially in the US, between the FDA and the USDA and the EPA, they will not try to make those match. And so it is not cost effective for a facility that's doing CGMP, that is making pharmaceuticals, that is making, food ingredients, to handle a product that doesn't fit into that regime. You could do it, but then of course it drives your cost up. And especially for products that are commodities or for agriculture, the cost just doesn't make sense for what you need to be able to sell that product for. So that's an issue. Another problem comes up when you've got a non-canonical host, right? If your host is anything other than E. coli or Saccharomyces or Bacillus, the facilities are going to say, wow, how do we make sure that everything in our facility is clean? If it's a spore former, especially if it's a spore former and it's not, something they're used to handling, they're like, oh my God, how are we going to make sure that, everything is clean for the next customer or our own production that's coming in after that. If you have a non-canonical microbe and maybe you haven't engineered it, but it's, a wild type host strain that came directly from the environment, you probably don't want to use antibiotics in that fermentation because it's probably not engineered, it's not resistant to standard antibiotics, right? And so we ran into this at Ag Biome, where we had a pilot scale facility that was otherwise very accommodating, happy to work with us. Everything was going fine. And then they found out they couldn't use antibiotics to control the cleanliness in their facility. And they were completely at a loss. Like, well, we don't know what to do now. So that was a problem. And then if you've gone even further, where you have a non-canonical host and you've engineered that microbe, and then you want to produce it at scale, and then you want to ship it, and you want to release it, and everything else, you're going to have a whole different regulatory regime. Your contract facility is going to want to see, copies of all of your permits. They want to know, how can they be sure that this microbe isn't hazardous to themselves, to their workers, to release. They of course don't want to get in trouble if they have an accidental spill of the stuff. So there's going to be a lot of additional regulatory documentation and just kind of getting everybody comfortable with that. And then there's going to be a new growth protocol. There's going to be new downstream processing. So like I said earlier, you got to build in extra time for the tech transfer around things like that. So all of this stuff, I think, kind of doesn't really surface in people's attention when they're talking about biomanufacturing until they're pretty far down the road. So I'm hoping that, this kind of a forum, we can, have some discussion about how to make that situation better. So as a consultant for Sable Fermentation, I can discuss a little bit about what Sable offers. Sable is a new company, CDMO, starting up here in the Research Triangle Park area. It's actually going to be in Wake Forest, North Carolina. And Sable is offering consulting and techno-economic analysis services. So I didn't even touch on TEA, but that's another area where making sure that a biomanufactured product is actually capable of making money. It can be produced economically in such a way that the manufacturer will actually survive. This is a service. There's other companies that are offering this, but we have a lot of expertise on the team, so we're offering that. And then a process development, optimization, scale up, spray drying, and analytical. All of that is covered by the equipment and the expertise we currently have on the team. And to dig into that a little more. So Sable has fermentation up to 200 liters currently. That's being installed, I think, next week in the facility. And then the plan is to go up to 25,000 liters with additional capacity on the same site. And then all of this other equipment is already also available and is getting installed. So the imaging equipment, the analytical capabilities, which it's pretty unusual for a company of this size to actually have all of that analytical, including the HPLC. So I'm pretty excited about that. That's gonna allow a lot of data collection of the output of the fermentations that a facility like this typically doesn't have. And then there's quite a lot of downstream capability, tangential flow for concentration or centrifugation. The tubular bowl centrifuge is kind of an unusual piece of equipment, again, for a company this size. That allows for a very concentrated cellular paste to come out, and that can be very useful, especially if you want to go then dry your product. And then the company owns a spray dryer, which is already up and running, and we can offer that service right now if you have things that need to be spray dried. So that's very exciting. This is really filling a niche here. As we discussed a little bit earlier, it's difficult to find tollers that will do all of this. And I think the team that Sable has available is really unique in this area. Also, Tony herself, the founder of the company, Tony Bucci, she had a long career at Ag Biome and before that at BASF. She's a very capable commercial executive. Her first hire was a fermentation engineer who I personally hired at AgBiome, so I was very pleased about that. Srjana has a lot of experience. She worked at Marone Bio. She was at AgBiome for almost 10 years. So she's done a lot of process development, a lot of scale up, a lot of downstream process development. They hired the operator of the equipment that they purchased. So that person knows all of that equipment inside and out, backwards and forwards. So that's very exciting. And we'll have a really capable staff actually on the ground in the lab. And then myself, I'm currently working with them as an advisor with all of my experience in doing this kind of process development, tech transfer, regulatory strategy, all of that. So very, very excited about what we can offer as a team. And that's my last slide. So happy to take questions about that and have a good discussion.
Fantastic. Thank you, Kelly. That's really interesting. And it's really great to see all the work that you've been doing and that Sable is doing right now. Selfishly, I would love to ask a first question to kick things off and then open the floor up. Both of these presentations have me burning with a bunch of questions, but I'll just ask one. And for me, it's like from the perspective of an academic thinking about a spin out or from a startup company, there really are such different incentives in the near term to be highly standardized to fit into what limited capacity there is for a CMO or CDMO. And then in the longer term, to sort of own your own process that, you know, as you can tailor, you then get that cost down or you get that product quality to where you need it to be. And so with these very different mismatches, I feel like there could be a lot of sort of pitfalls where if we take a very simple one, like mode of operation, maybe in the beginning, you're optimizing everything and your strain around batch mode fermentation. And then longer term, maybe you're like, okay, a longer continuous campaign might make sense when I have my own equipment to do so, but maybe you don't have the genetic stability that far out or, you know, all these other considerations. Do either of you have some advice or some considerations for someone who's thinking about that maybe being dragged in one way or really good at for a CMO, but then they never make commercial viability? Like how to think about that?
Rasmus, do you want to tackle that one?
Yeah. I mean, at least I would say that that if you're betting on the wrong strain, I mean, all the time you use on that strain is eventually lost. I would say it's a blind alley, so you need to make the right choices as early as possible. Right, a lot of our companies are people who started out as molecular biologists in the lab because they knew how to make strain construction, but they never had access to industrial strains, right? So it's a very, very difficult decision for them to abandon their own strain program. Quite often it's their own capabilities. It's potentially having to even fire people in the company because you may not need those competences anymore. You've made promises to investors that you're going to own your production technology and so forth. And we cannot offer that, right? We're based on licensing the Novonesis technology. So I think, I think it's not a good advice to try to get as far as you can with your own if you know that it's never going to be good enough. Of course, you may need to take it far enough to make small samples to do application testing and so forth, but really trying to start scaling a technology which is never going to be economically feasible, that's a waste of money.
Yeah, I would echo that, and I think that's another area where the techno-economic analysis is so important. You can use those mathematical models to help you project into the future. You know, is a particular host or a particular production pathway, metabolic pathway, are those eventually going to be scalable and be economical to produce with or not? And you're absolutely right. People do kind of get a little bit attached to their own their own strain, their own technology, and so I think you have to go at it with a very clear eye towards, you know, what does this really look like in the future, and those models can help. What constitutes a minimum viable product in a biological process is a matter of intense debate, right? Is it a small production sample? Is it a microbe that's been scaled up to a certain scale? Is it actual customer verification about the product? I think it kind of depends on the industry that you're trying to get into. Somebody's got a hand up there. Paul?
Hi, everyone. First, thank you to both of you for very nice presentations. I have maybe 2 questions, if I'm allowed, or one question and one is more a naive question. But all of that is great. As an end user, a company who may buy some of these ingredients, because we are not in the business of producing these ingredients, so we're going to buy. Let's say we have potential supplier of these ingredients. I'm left with maybe 2 hurdles that are very lightly addressed so far, or at least I've seen for every conversations I've had. There is very little conversations around targeting the right protein functionality, and how can we be sure that the technology that you guys are presenting are really making the right functionality of the proteins. And if you talk to a few professors around, they are saying that's the biggest hurdle today. There is lots of dreams promised, but how do you get that? And then the second is the CO2 footprint analysis. Lots of nice dreams also, 90+% CO2 reduction. But when you talk to some engineering companies, they say, these guys are dreaming. It's never going to be like this. And hearing that there is no production capabilities or everything is being scaled up, how can we really be sure of the CO2 footprint? So for me, these are the 2 hurdles for these ingredients to be mass market. And then the second one is maybe more on Kelly, but it's not the first time that I see people like this. They talk about fermentation capabilities in liters, but as an end user, I don't care liters. I want to know how many kilos of powders can I get after the fermentation. So it would be great if somehow this area stops talking about fermentation volume of their fermenters, but talk about yield or kilograms of powders that I get after spray drying.
Yeah.
I'm going to let Rasmus tackle the first one, and then I'll tackle the second one.
Yeah. So to the first one, for the LCA calculations, I think you don't really know until you have a dedicated factory, which is made to produce a specific protein, right? Because then you have the cheapest possible factory set up, and it's optimized to be as efficient as possible. Then you really know. And the numbers we are throwing around, I think they may be off, but they're not totally wrong, for sure. But it's when we talk about milk protein, at least with our strain, it's not been scaled above 10,000 liters yet. So we don't really know. Your other question, it slipped my mind. Could you just repeat the first question?
On the protein functionality.
Yes, the functionality. From 21st Bio's point of view, that's specifically the responsibility of our customers. We will help make the strain and the production technology. They need to feed back information to us if the protein is not working correctly. For example, do you have truncation of the N-terminus, or that changes functionality, or do you have glycosylations or phosphorylations or something that are off? That needs to be tested, and the information needs to be fed back to us so we can make the proper amendments. So we've decided that we want to stay out of the application testing because our heritage from Novozymes tells us that that's just a huge task, and when you're operating across many different fields, that's a big and lengthy investment. But I think all of our customers in the milk space, I mean, that's really what they focus on, making the proteins work in different aspects of formulating food products.
Okay, so then on the question of the actual kilos of output from a given volume of fermentation, I think you're absolutely right. One of the reasons that we end up in this situation is because people with my background, the fermentation engineers, if you ask them, well, how much, what kilos of output can I get from a 200-liter fermentation? What they'll say is, it depends. So I think the TEA can help a lot with that, right? You can use that techno-economic analysis to say, okay, with this metabolic pathway engineered into this microbe that grows up to this level, then you can expect to get, this range of kilos of output, especially if you use our spray dryer or whatever, right? You can easily model that if you have a little bit more information about the process. But with that said, I think it would behoove us as an industry to start talking about kilos of output, right? Say, and in a typical E. coli production process, that means you get X amount of protein out of it, with all of those caveats that every process is going to be slightly different.
Yeah. Matt, I have a customer call, so I need to hop off.
Yes, of course. And I wanted to say, it feels like we're just picking up steam, but I understand we have to end at this point. So thank you very much to both of our guests here. If open to it, happy to make some connections. So happy to facilitate and discuss on both sides to see what happens if we want to keep going out of here. But thank you so much, everyone, for your time. Really, really appreciate it. Really,
happy
really
to hear
interesting.
from anybody who wants to talk.
Thanks.
So.
You're welcome.
Thank you. Bye-bye.
Thank you.
Fantastic.
Okay. Bye.
Cool.